COVID-19 therapies made from antibodies are often given to patients who are high risk of severe illness and hospitalization. But virologist and immunologist Dr. Michael Diamond at Washington University School of Medicine in St. Louis wanted to answer nagging questions about whether such antibody therapies retain their effectiveness against variants of the virus that causes COVID-19.
If there was a drug that was available as an antibody therapy by AstraZeneca, AbbVie, Vir or Lilly or Regeneron, what would happen in the context of new variant viruses? We already knew from some studies in cell culture that some of the antibodies might have liabilities against some of the variants, meaning that they might not show as great efficacy. But they had not actually been tested in any animal model to be able to assess whether they could still protect.
The professor of molecular microbiology and of pathology and immunology wanted to know whether a single antibody therapy or dual antibody combination therapies available through emergency use authorization or part of advanced clinical trials would still be effective against new and emerging variants. So Dr. Diamond's lab put the therapies to the test against variants in animal models.
Test them against a panel of different viruses, SARS-CoV-2 viruses, including variants of concern like the beta variant, the alpha variant, and ultimately study them in the context of others such as Delta variant.
Many, but not all, dual antibody therapies made from combinations of two antibodies proved effective against a wide range of variants.
I say most, in particular, the AstraZeneca, Vir, and Regeneron combinations did quite well against all the variants. Antibodies that bind different sites may actually work together to get what we call synergy. So if you take one antibody, it has a certain activity. If you take another antibody, it has another activity. If you add the two of them together, you can either get additive activity or you can get even better than that, or what we call synergy, such that when the two antibodies are combined, they actually work better than either of the components of the combination.
The research suggests that COVID-19 drugs made of two antibodies often retain potency as a therapy against variants and appear to prevent the emergence of drug resistance.
If you have two antibodies, you have to have almost simultaneous events to get resistance to both components. And that turns out to be evolutionarily very challenging for the virus.
The initial study took place before the highly contagious Delta variant became a problem. More work needs to be done testing the therapies against the Delta variant in animal models. Diamond says the work done here helps the FDA make decisions.
Our data sort of highlights to the FDA and to others, hey, you need to evaluate this to make some decisions, whether we need to limit the indication for certain antibodies to certain strains, or we need to think about broadening the coverage.